Okay. Can you wait a bit?
Okay.
Yes. Not a strong protocol, don't worry. Good morning, dear students, dear jury members, dear audience following us online. It is an honor and a big pleasure to open the very first session in this room, the Whale Room of the 7th edition of the IMBRC Annual Symposium 2024, where we have cohort 2022 students showing us their master thesis work. As the main coordinator of the IMBRC, I couldn't be prouder to be standing here and welcome some of my students. So it's my pleasure to call into the stage Patricia Navarro Gonzalez, who's going to tell us about mixed up in fishy business, using drift models and correlation with fishing activity to evaluate the strandings of common dolphins, Delphinus delphis, in Irish waters. Patricia, you have 15 minutes to present, and the jury has up to 15 minutes to have questions. Please, Patricia, the floor is yours.
Hello everyone, like Luana said, I'm Patricia and I'm here to present my thesis project. First of all, I will introduce you the issue. So bycatch, marine predators are large organisms with wide range of movements, and therefore they are exposed to a wide variety of stressors. Those can be from causing mortality to disturbance. In the fatal group, we can find bycatch, which is all unwanted catch of fish and other marine vertebrates of no commercial value, and this has become a source of conflict for marine mammals and the fishing industry. In fact, it has been highlighted as the main stressor for cetaceans in the Northeast Atlantic and has negatively impacted their population's health and the individual's welfare. The two most impacted species in this region are the harbor porpoises and the common dolphins, and they have been impacted by different gears such as gillnets, quercenes, and the different types of trawling targeting both demersal fishes and shoaling pelagic species. But how do we quantify that bycatch? Well, under the new regulations of the European Union called the Technical Conservation Measures, for all fishing vessels over 15 meters, member states are required to have observer programs on board, and they are the ones reporting the bycatch numbers. However, the current bycatch numbers that we have are unreliable, and this is because inconsistent application of the measures by the member states, and because the programs are inherently biased by the observer effect, skewed to different seasons and gears, and have undersampled the different fleets. Rather than relying on the observed cases of bycatch, we propose to quantify bycatch through the past interactions of the carcasses with the fisheries through stranded carcasses. And strandings have been a key source of information for marine mammals, ecology, occurrence, life history, and anthropogenic stressors. And combining strandings with other disciplines in a way that we can track the past trajectories of the carcasses could provide solutions for bycatch. Those systems are called the Lagrangian or drift models, and they are oceanographic models that track particles using different oceanographic variables. They are quite a novel technique, but they have been proved useful in several marine species such as dolphins, whales, turtles, fish, and hydrozoan species. Therefore, in this project, we chose the common dolphin as the study species for the project because of the increase in stranding in the recent years, despite their consistent population, and they're unfavorable to an adequate conservation state for the Northeast Atlantic, where a rise in the human activities is predicted to occur. In this project, we aim to quantify the bycatch numbers of common dolphins in Ireland through external necropsies on the carcasses, and identify the special correlation between common dolphins at sea and the fishing effort in Irish waters. As you can imagine, the stranded data was not usable right away, so how did we get from the stranded data to the spatial correlations and the statistical power in this project? I built a roadmap and I will now guide you through it. First of all, for the strandings, for the drift models, I needed to know where the carcasses ended up, when they stranded, whether they stranded through bycatch, and any other oceanographic variables related to their time of death. So first of all, I collected any type of strandings data related to the carcasses with photographic evidence from the Irish Whale and Dolphin Group. Then necropsy reports from opportunistic sampling and from the National Parks and Wildlife Service and from the Marine Institute. Also, oceanographic, fine-scale oceanographic model for the Northwest Atlantic, and commercial fishing effort from the Marine Institute for the same period. Then, I should have put a disclaimer for these pictures, but then I processed the pictures of the carcasses using the most up-to-date guidelines on necropsies. So I used the external features that I could find on the carcasses to estimate the time of death and cause of death. Time of death, I classified it in 5 different categories that is consistent with the literature. And for the cause of death as bycatch, I identified the most common external features and then classified them to certain or possible bycatch. Then I combined the picture analysis and the necropsy reports that run the drift models on the carcasses. So for each stranding event, I ran 1,000 particles backward from their time of stranding to the time of death based on the different categories of the composition. And that is a simulation of how the drift modeling looks like. Then I ended up with 1,000 particles per stranding event, and I aggregated them using the kernel density from ArcGIS. And for an easier visualization, I rescaled the data into percentages. And that is what I call the hotspots. Regarding the statistical modeling, because the data are spatial data, I ran Moran's I autocorrelation, and the data was in fact autocorrelated. So I included the spatial variables, the longitude and the latitude, as an interaction in the general additive models, and all the other fishing variables related to the gears and the different flags. And because the fine scale distribution of the data, there was a high abundance of zeros and low values, so I log transformed the data before putting them in the models. Up to now, that was the methods, but what did we find out? Well, regarding the bycatch assessment, 202 out of the 1,160 dolphins that I analyzed between 2013 and 2023 were confirmed or suspected to be bycaught. Out of those, in the left-hand part of the graph, those are the ones that came from necropsies, 24 animals, and the rest, that other part, are from the pictures. And as you can see, the pictures, I don't think it's very visible here, but there's a dotted line that goes in the blue lines, and those are the strandings distribution, and the black thick line follows quite closely the stranding distribution, which could mean that a lot of the strandings could have been bycaught. Then, as for the decomposition, 20 to 50% of the animals were in good or fresh to slightly decomposed conditions. However, the boundary between both categories was quite blurry, and this is because high scavenging of the bodies despite an overall general good condition of the body. This has been found in other areas in Europe, and it highlights the lack of availability of food resources for the scavengers during winter. And because I didn't have access to the depth or the angle of the marks, it difficulted finding net marks in the body. The implications for this is that actually not all fishery interactions result in net marks in the body. And so that is when necropsies come in useful, because they can find other internal clues in the body, such as drowning, fish scales, and subdermal bruising that are not so visible with pictures. However, it is financially impossible to run a gross or even full necropsy on every single animal that strands. Therefore, this new technique of external necropsies was still quite successful, since it identified 202 bycaught dolphins as opposed to the 7 reported bycaught dolphins in a similar period. There was some variability in the trajectories of the common dolphins in the drift modeling, and there was a clear differences between the dolphins drifting further offshore and dolphins staying closer to shore. This is the map of the aggregated points, and we can see that there was a lot of hotspots all around Ireland for mortality events. What I mean by hotspots is not the actual true numbers of by caught dolphins at sea, but rather particles by area, so they are a proxy of the mortalities or the location of the mortality areas, and the map doesn't have any statistical power. We can see that there's a lot of numbers in inshore waters, and they are located mostly by the counties of Galway and Clare, Kerry, the whole south coast over there, up north by Dublin, and up north by Sligo and Donegal. And the offshore waters, they are in more like pinkish colors, they have lower densities, but there were still some areas of significance, and those were located in the South Seas, in the southeast, in the south coast, in the southwest between the island and the Porcupine Sea Bight, in the Porcupine Bank on the northwest, and the northwest up the continental shelf. As I previously mentioned, there were disparities in the homogeneity of the trajectories, and also there were difficulties in the assessment of the pictures. So this meant that relying only on necropsies versus the picture analysis meant that with necropsies we would have had only very localized hotspots, whereas with the pictures they covered a much wider range. And animals that were identified as fresh animals could have traveled further offshore if in fact they were slightly decomposed. So that is fresh and that is slightly decomposed, and we can see that they go a little bit further offshore. Regarding the spatial co-occurrence, so the co-occurrence between the fisheries and the hotspots, we could see that the overlap was greatest for the bottom trawling, those two over there, followed by the gillnets, the purse ends, and the pelagic trawling, and the intensity was greatest for the last three. The spatial location of these overlaps for the bottom trawling, they covered all around Ireland, but they were mostly offshore. For the gillnets, they were restricted to the southwest. For the purse ends to the south, and the pelagic trawling on the northwest, with some inshore hotspots. Some inshore areas, sorry. Overall, it is worth noting that there is a lack of overlap in inshore areas despite most of the hotspots being in inshore waters. I described the spatial relationship before because not all the gears and flags had a significant relationship in the graphs, despite almost 17% deviance. Most of the gears were Most of the gears were significant, and they're highlighted by the asterisks. And only the purse ends, those two over there, and the international gillnets were not significant. Their relationship was positive for the international pelagic trawling and the Irish gillnets, and negative for the Irish pelagic trawling and the international bottom trawling. And the rest, the relationship was almost flat, as you can see by the purple line. Drawing from previous studies, the risk of bycatch was suspected to be highest for the pelagic trawling in the northwest and the gillnets in the southwest. We did see a similar overlap in our maps, but not all the gears and flags were significant in our model. At a wider scale, the marine mammal bycatch indicator from OSPAR also highlighted a very similar relationship that for common dolphins in the Northeast Atlantic, pelagic trawling and gillnets have the highest bycatch numbers overall. Purse ends for the whole region have generally a very low risk of bycatch, and they pose a risk mostly in very localized areas, which could have also been the case in our study, and that's why the relationship was almost flat. Because of the consistency between these previous studies and the overlap maps that we found, that led me to think that perhaps it was the landings composition of the fisheries that had that role at play. And in fact, between 2014 and 2018, the pelagic fisheries in Ireland targeted blue whiting, horse mackerel, mackerel, sprat, herring, and albacore tuna. And across the board, these are either common prey species of the dolphins, so pelagic shoaling species, or predatory fishes that have prey in common with the dolphins. For future work and recommendations, based on the discussion and the conclusions that we had, we recommended a more in-depth investigation into the influence of the bycatch assessment, the decomposition of the body, and the certainty of the assessment on the co-occurrence, because this would lead to more accurate maps. An improvement in the statistical methods by including the spatial variables as a mixed effect instead of an interaction, the temporal factors, which we didn't include, and the landing composition would lead to identify the fisheries that pose most risk for the common dolphins in Ireland, and also identify what types of targets are the ones at play. Also, in the European Union, generally, acoustic deterrent devices are the mitigation that they recommend in general for everything. However, there is a move to more spatial temporal closures because they have a much higher success in pilot studies. And this could, after identifying the fisheries with the most risk, this could be a solution for the Irish waters. And as we saw with the difference between the overlap map and the hotspots map, there was a lack of overall data on fishing, inshore fisheries efforts. And this is a problem because in Ireland, inshore fisheries are 86% of the fleet. Finally, to conclude, this was the first time drift modeling was applied in Ireland and with quite successful results. The citizen science photograph proved to be a successful way of analyzing bycatch as well as a cost-effective technique. Yes. There were inshore and offshore hotspots of bycatch identified, and there were significant overlaps for demersal and pelagic trawling efforts, and significant overlap for gillnets. And across the board, this could be explained because they targeted either shoaling or predatory fish. So with this, I conclude my presentation. Thank you very much.
Thank you very much, Patricia, for this very interesting presentation, ambitious project. I would like to give the word to our jury members. Who would like to go first?
I can start. Thank you for this presentation. I really liked it. What about the number of baits that are stranded? What's the total number in Ireland? Do you know per year or?
Actually, I did have another map for that, but I don't remember the numbers. But so I only analyzed 1,160, and that was from 2021 to 2023, because that's when they started asking the volunteers to take pictures of different angles, and so only those pictures were reliable. But the numbers were much higher before, so I think, I don't know if you can see this map, but because it's quite blurry, but so this is 100 and 200, and like for example for February in general we get around 300, and then so the numbers could be between 2,000 and 3,000 more or less, I guess.
And this particle model that you use, what's what's it, is it driven by? Is it wind or currents, or what's behind them?
Yeah, so the model in general, it includes the whole water column. So it includes the tidal currents and the normal currents. And you could also apply wind drift, but I didn't have the data for that because only the current data was 14 terabytes. So
but aren't they floating?
Yeah.
So the wind could be really, really important.
It could be, but they did an experiment in France in 2012, and they saw that the dolphins, as long as it was before 20 days of after the time of death, they would float or partially float, so they would be within the first layer of the water column.
to go on.
Okay.
So thanks for this nice presentation. So can you tell me again what is your driven hypothesis of the work?
And you repeat, sorry?
Can you tell me which is your driven hypothesis
My
for
main,
your work?
my main hypothesis.
Yeah.
Yeah, so we were expecting more common dolphins to be offshore than in inshore waters with the drift modeling, and we also expected, as I mentioned in my discussion, pelagic trawling and gillnets to have more effect because they have been problematic in Ireland. So the bottom trawling was a surprise in the models, and then finding out most of the inshore hotspots was also a surprise.
Okay. Then do you know the limit of your model?
The limit of my model?
Yeah, or the limit of your methodology?
Yes. The limitations is that for the drift modeling, at least, I only include the first layer, and because this drift modeling was tested in France with the experiment, it could change, and actually the dolphins could sink a little bit more in Ireland, so that could have been another problem. The necropsy that I did, there was a lot of variability in finding the net marks and finding the different compositions, so that could have changed my map as well. And for the statistics, the data had a very strange distribution. And so I did what I thought was the best model, the general additive model, but because it didn't have any temporal variables and not many error variables, I think it could have limited the results, yes.
Then, okay, there is some previous methodology that has been used to address such bycatch question, and you are aiming to develop or to use a new one, maybe more complete. So can you comment on that, and can you tell us what would be the strength of using your methodology?
Yeah. So the previous research on bycatch has been mostly based on the risk. So they take the distribution of the dolphins and then the distribution of the fisheries and the different temporal factors, and they say that this fisheries has the most risk. But overall, like I mentioned, in Ireland, for example, they have only ever reported 7 common dolphins, but we know from the Northeast Atlantic that actually in Irish waters there's a very high risk of bycatch, it's the second region for the most risk in Ireland, in Europe. And so there's no research on the realized risk, and so the distribution of where the animals are bycaught. And because most of the research relies on the animals that they take from the fishing vessels, there's not enough data, and with this at least you can get an approximation of where the risk is happening, where the bycatch is happening. And you can actually, if you do probabilistic analysis, you can get the amount of dolphins that could be bycaught at sea. So for me, it was just the densities as an area. But this person that did it in France, they included the probabilities and also included all the animals that could have been stranded but never stranded to get a proxy of the animal bycaught. And it has been used by ISIS to identify different mitigation strategies and how different mitigation strategies could have the different levels of success.
Okay, so then with your set of data, so what will you do next?
So I would include more temporal variables, and I would include the different targets of the different fisheries, because even though pelagic fisheries, for example, are suspected to be bycaught is more often, it's not all over the overlap, it's in specific regions. So they'll also having the spatial variability, the temporal variability, and the prey is a much more powerful technique. So I would do that, and I would probably do the external necropsy in a more standardized way. And by that I mean that I don't mean that it wasn't standardized before, but that because there was a lot of variability, now knowing that, I would change the different categories.
Yeah.
No, we are running out of
Can I
time.
ask
So
one
Yes.
question? So now at the conservation level,
Yeah.
So what you would do with such data?
Well, it would be exploring the different mitigations because in Ireland, the only mitigation required is to have acoustic deterrent devices, but they barely work on common dolphins. They work mostly on harbor purposes. So, yeah, there's nothing much being done otherwise. Thank you.
Okay. But Patricia, and I'm also gonna ask you one or two questions. Most of my questions have been addressed to our jury. How would you go to try to elucidate more about the ecological behavior of your target species and to try to elucidate the relationship about who is eating who and what is influencing actually the presence of the dolphins closer to the shore or offshore?
Yeah, so the common dolphins actually change their distribution depending on the season because during the summer they're in the calving season, and so they need more prey intake and they go a little bit further offshore. So that's why I wanted to also include the temporal occurrence because the risk changes especially between different seasons. And there's only been prey studies done in the late 90s and the early 2000s, and their spatial distributions has changed since then, and so the preferred prey has probably changed since then as well. And some of their species that they had mostly preyed before, they have lower densities now because they have been overfished. So yeah, that would be something to take into consideration.
And did you thought about how do you convince the policymakers to adopt new methodologies or enforce a strategy that, from what you just told us, is actually not even being taken care of the common dolphins in Ireland?
Yeah, well, in France, that took 10 years from the first time they developed this method until it was actually used by ISIS. And it's just improving the methods and showing that actually there's a huge difference between the numbers the government is reporting and the numbers that are realized being caught.
And but you do have any idea? How do you really convince? Do you need to convince how do you convince the policymakers? Which kind of way can you put some pressure on them to address this matter? Do you have any ideas?
That's a good question. Well, the thing is that most people that work in this field, they don't work that much with the policymakers, but rather with the industry. It's like a bottom-up approach, and that helps them get more realistic numbers of bycatch, but also have mitigation strategies that work for the fishing industry as well, because they're the ones being impacted. For the policymakers, as long as they already know that it's an issue, but the numbers that they have just don't represent the issue, and so they've been saying for decades that there's no risk of bycatch. So I don't really know how I could convince them that what they already know.
But for you, what is the most important part of your research, or what would you like in the future, even if it takes 10 years, to see happening?
The actual realization of the mitigation strategies, yeah, based on the maps.
To protect the species?
Yes.
Because, for what you say, there's a difference between the goals of the scientists, natural scientists, and the industry makers. What are their goals? It's because of their product.
Mm-hmm. It would be to reduce the mortalities of the species.
Yeah. So maybe we need to just bring more awareness to the general public, right?
Yes.
Okay. Thank you very much.
Thank you.
Patricia.
No, it's not. This is Margaret.
Before I introduce our next presenter of the day, I'd like to properly introduce our jury members. My apologies for that. Aldina Amir from Universiteit Côte d'Azur and Per Alberg from University of Gothenburg in Sweden. It's my honor to call into the stage Margarita Medina, who's going to tell us about long-finned pilot whales, Lobicephala melas, in offshore Irish waters, insights into open-source software techniques for whistle detection, habitat use, and shipping noise impact. Margarita, you have 15 minutes, and also I will signalize to you when you are through, and then 1 minute.
Yeah, thank you. Okay, so welcome everyone. My name is Margarita, and I've done my thesis in bioacoustic, focusing on assessing the offshore distribution of long-fin pilot whale in Irish waters. For this purpose, I tested a new open-source software technique for whistle detection, and I investigated the potential shipping noise impact on this species. The data that I've used for this study were sourced from an acoustic monitoring initiative happened in 2015 and 16 in offshore Irish waters, called the Observe. And I would start giving some background on the species of interest, Lobicephala melas. Those are odontocetes belonging to the Delphinidae family. They inhabit mainly offshore waters. They're considered deep divers, and they highly rely on vocalizations for various biological functions. Their vocal repertoire is considered one of the most intricate among all mammalian species. They can utilize different types of calls, and within these calls, there has been observed a huge variability of call structures. To simplify, we're going to focus only on the whistles or tonal calls. And here again is a simplified scheme of the current status of knowledge regarding their frequency range with a comparison with other smaller odontocete species on the top and other larger odontocete species on the bottom. Among the conservation concerns, there's for sure anthropogenic noise. And while seismic surveys and military sonar has already been studied as a potential threats to the species, commercial shipping noise has not been yet investigated. And it is particularly crucial to study commercial shipping noise impact in Irish waters, since in the last half-century there has been a surge in 20 decibel in ambient noise attributed to this human activity. Moreover, the species of interest is listed as data deficient in the Atlantic, meaning that little is known about its distribution, and is also reported as one of the most frequently stranded species along the Irish coast. Sorry. Okay, to fill the gap of knowledge regarding those elusive species, in the past literature, mainly static acoustic monitoring surveys has been employed, with the bottom-more fixed autonomous recorders gaining more and more popularity due to their cost-effectiveness and suitability for long-term monitoring in remote and challenging areas such as the Atlantic offshore waters. One disadvantage held by this methodology is the need to deal with big data analysis, but this could be overcome by the development of automatic detection methods. And I've seen that in the recent literature, mainly proprietary MATLAB-based tools were utilized to develop algorithms and tools able to discriminate these species vocalizations. And only in a few cases, open source software such as Vanguard was utilized, and mainly to train a detector for this purpose. My research objectives were to test a new open source software technique for G-melas whistle detection with no requirement for detector training, meaning that I aim to use the Vanguard bioacoustic software utilizing a predefined, already available algorithm called the whistle amount detector, which is a tool that was already being used for other dolphin species but never before for pilot whales. Then I aim to, with those data, to investigate the offshore occurrence in Irish water of this species and to examine the impact of shipping noise. Okay, moving on, here's a map showing the location of the mooring site that I've chosen for this study, which is placed in the northern edge of the Irish continental shelf at almost 2,000 meters deep. And here you can see on the left a scheme of the mooring system itself, and in red is highlighted the shrouded cage in which is contained the AMR device. which is the acoustic recorder provided by JASCO Applied Science, a well-known bioacoustic company, for this purpose, and was set to record at 2 different sampling rates for a total duration of 4 months, from May to August 2015. The shipping noise data were extracted in the context of the observed study by the JASCO team using their own bioacoustic software, PAMLab. And first of all, the sound pressure levels of the signals produced in the frequency range between 40 and 300 Hz were extracted, since this is the frequency range commonly attributed to mid and large size vessel. Whereas the shipping tonals also were extracted, and those are the narrow band tonal sounds produced by the propulsion and other rotating machinery of the vessels. Okay, moving on. As part of the observed project, the JASCO team was also responsible for marine mammal vocalization extraction. And in the realm regarding GMLS whistle, the JASCO team utilized the PAMLab software to develop a call sorting algorithm specific for the species that resulted in the threshold values here listed on the left. And I aim to utilize those tested values in Vanguard. And I'm not going to go in detail on the configuration settings, but I'm just going to say that to do that, I needed to employ a so-called audio data acquisition module, which is used to upload the sound files of interest and set the needed calibration parameters. Then I needed to link that to an FFT spectrum engine module, which is basically a mathematical algorithm that converts a signal from its time domain to its frequency domain. And this was in turn connected to the whistle amount detector, the predefined algorithm that I aim to test. The parameters that I've used in these configurations were sourced from a pilot study that I performed on a subset of data, taking inspiration from previous studies that has been done on other dolphin species on the whistle, using the whistle amount detector, as well as some other values that were used by JASCO in their own bioacoustic software, and some other values that were instead suggested by Vanguard. Okay, those Vanguard detections were then converted into a presence-absence per hour format, and a manual validation was performed in RavenPro, inspecting 1 whistle per hour. And with the manual validation results, I computed 3 performance metrics: the F-score, the Matthews correlation coefficient, and its normalized value, which are widely used metrics in binary classification evaluation. Okay, the validated whistle detections were then analyzed in relation to the shipping noise data provided by JASCO and other potentially relevant environmental drivers. And I've done that in our studio using a generalized additive model. And the best model resulted in the one having as covariates the 2 shipping noise data, and as well as chlorophyll, sea surface current, moon phase, and hour of detection. Okay, moving on what did I obtain. Here in the slide you can see on the left a bar plot showing the comparison between the presence hours detected by the Vanguard detector versus the presence hours validated, so resulted from the manual validation that I performed, the actual presence hours. Here on the right, we have instead a bar plot showing simply the counts of true positive, true negatives, false positive, and false negatives to give an idea of the total amount of correctly classified hours versus the incorrectly classified hours. And from this data, we can see that, first of all, the Vanguard detector seems to slightly underestimate the presence of pilot wells in the area, identifying as presence hours just 30% of the total investigation time, whereas the manual validation reported that almost 50% of the investigated time were actually presence hours. Anyway, however, looking at the right plot, we can see that the detector was able to correctly identify and classify 80% of the hours. And this resulted in the following performance metrics, and I just wanted to bring the attention to those 2, the F-score and the normalized MCC, and they're both showing an overall performance of the detector of almost 80%. Then, moving on to the modeling results, we can see that from the partial plots, which are basically representing the partial effect of each smooth third included in the model on the response variable with the corresponding confidential interval, we can see the some pressure level of the shipping noise seems to actually have a negative effect on the response variable, so the whistle detection, whereas the number of shipping tonal detections seem to have an overall positive effect. And moving on to the environmental variables instead, both concentration of chlorophyll and sea surface current, they both showed a slightly positive effect on the response variable. Regarding the categorical variables inserted in the model, for moon phase, just one level seems to be significant. And we're talking about the new moon phase, so it seems that during this phase there has been like a higher probability of whistle, long-fin palliative whistle detection. And instead for hour of detection, overall this variable seems to be significant, showing a slightly decrease of probability of whistle detection throughout the day, so with more detection during the first night times. So overall, I can say that this study showed the potential efficiency of the whistle hormone detector in extracting Globicephala whistle. And we can see that from the performance metrics that I calculated, and comparing that with the ones that has been calculated by the JASCO team for the PalmLab detector. And even though my values seems to be even higher, so suggesting like better performance than one of JASCO, it doesn't mean that my detector performed better, but it's just a matter of like how the manual validation was performed. So in my case, for the purpose of my study, was performed per hour, whereas the JASCO team has performed the manual validation per sample, meaning approximately every 6 minutes. Okay, one big limitation of this part of the study was the small sample size used in the preliminary Vanguard configuration test. So future studies should for sure employ a larger sample size for this pilot study to gain more specific insights into the impact of each parameter, enhancing the overall final configuration setting. And from the habitat modeling instead, we can see that all the environmental variables selected actually suggest the importance of the area investigated for the foraging activity of this species. In fact, the positive relationship with chlorophyll can be explained by the fact that higher levels of chlorophyll has been linked with a higher abundance of deep sea cephalopods, which are the main prey of these odontocytes. Moving on, sea surface current can affect the foraging behavior of these odontocytes, affecting the nutrient movements along the water column in the so-called shelf zone front. And concerning hour, the negative effect found aligned with the well-known habit of this species to perform foraging dives at night, aligning with the vertical daily migration of their prey. And just for moon phase, actually the correlation found in my study was not found in previous ones, so I think further studies should be done to investigate better this correlation. And concerning that vessel noise impact, the model at first show a pretty contradictory relationship, so it means that it seems that the sound pressure level could negatively affect the whistle presence, the long-fin palliative presence, whereas the shipping tonals could positively affect that. But this could be explained by the fact that when the vessels are close to the recorder, due to the cavitation noise of the vessels, fewer shipping tonals are detected because it's harder to get on the spectrum these narrow band tonal sounds because the sound pressure levels are too high. Whereas when the vessels are far from the recorder, more shipping tonals are detected due to lower sound pressure levels. So it makes sense to have a greater probability of pilot rail whistles with low sound pressure levels and higher shipping tonals, because this could mean that we just have a greater probability of detection when the vessels are far. Overall, we need to interpret the results of this model with caution, because my model actually had a deviance explained pretty low, and this could be due to many factors. But for sure, future study, in order to enhance this modeling part should maybe change the response variable format in maybe number of detection per hour, not presence absence, to get more quantitative insights. Then additional Moore insights and multi-year recordings should be maybe included to overcome the lack of spatial insights and limited temporal range proper of this study. And lastly, I believe that incorporate heat detection data could actually better describe the presence of these species along the water column, so not just having the whistles. I can say to conclude that by performing this study, I provided the initial guidelines for an alternative, efficient, accessible, and reliable extraction of G-Melas whistles. And then through the habitat modeling analysis, I believe I shed some light on the offshore distribution of the species, aligning with the European Habitat Directive conservation objectives for the species. And lastly, studying the shipping noise impact, I maybe possibly provided new evidence to support the Marine Strategy Framework Directive investigation on the rising underwater noise. Thanks for listening.
Thank you very much, Margherita, for this interesting presentation. Adil, would you like to start now?
Yeah. Will you?
Yeah. Okay. Thank you for this really nice presentation. How deep did you settle your device?
Well, unfortunately, I didn't do it myself. I couldn't get involved in the project, but it was set at 2,000 meters deep.
2000. So do you think it's the optimized depth for such study?
Actually, it could be a little bit, because we know that those odontocysts can dive like over 1,000 meters deep. So yeah, it could work. But even like a little bit shallower, probably we could have also other good insights. For sure, not deeper than 2,000 meters.
Okay, then regarding the data collected, so from how many devices they were coming from?
Just one device.
Okay, so then how can you envisage optimizing and improving your study?
Well, for sure, we would need, again, to for sure, include in the model more insights. Of course, due to time constraints, I could just analyze the data for one single device, especially because the manual validation process was really long, and it was really needed to have validated whistle detection to provide reliable data. So yeah, for sure, we suggested I would focus on additional more insights and multi-year recordings to be able to identify also seasonal trends, because having just 4 months, I still actually identified a peak in July, which was also fine in other studies. So it was interesting to see that overall I also find a kind of fraction of seasonal trend, but for sure that's needed to really know and get more insights into the distribution of the species.
Then regarding the depth of the device settles, so do you know, okay, you say that you mentioned, correct me if I'm wrong, but that the weight, I mean the surface noise can affect somehow what you are recording with your device. So do you know what the absorbance of this noise within the water? Or
actually, I don't know how the sea surface actually could affect, because having the device so deep is actually helping. I also it's not really connected anyway to the sea surface, because it's really you can retrieve it through a acoustic release system. So it's just really at that depth. So I would say that the sea surface is not affecting that much, but I don't know the details of the.
And the contrary, so in terms of geology of the site, you put your device in. Do you know if there is any, I don't know, movement noise that can really affect?
I believe that's probably a factor, but yeah,
You never asked to any geologist, I mean, what is the geology of the site and the dynamic of the site?
Actually, in the observed, because I gain all those information from the observed final report. So it's the final part of the project where I've taken the data. And I haven't seen, so according to my knowledge, I haven't seen any mentioning on like this aspect, but I'm sure that there could be maybe some effects due to the seabed. You're welcome.
Thank you, Tim.
Yeah, I wonder a little bit about the scale here. How far away from the moorings can you actually hear a wave?
Well, it has been calculated. I have a backup slide for that.
Yeah, okay.
Like in which I'm just okay. So this is just an example of like because I think you meant like the detection range. So the detector was actually calculated by the observed statistician, and the long-tail pilot whistles were detected from a maximum distance of almost 30 kilometers. Whereas, like for instance, for other baleen species that have like a lower frequency localization, the detection length was larger. But for my species, it was more or less like 30.
Okay, and then you have the picture of the ship here. So, how far away can the ship be to affect this? So when you say that the ships, they do have an effect, but how far away will they be?
This is definitely a thing that should be studied more in depth because, of course, like since this is the first time, according to my knowledge, that this shipping noise effect was investigated. I feel like, in my study, I could not I'm sure that there has been calculation on again, like the detection range of the vessel signal. So probably knowing that information, we could also know more or less the position of the vessel from the recorder and also the relative position of the long-fin pilot well detection from the recorder and gain more insights. But in this study, I really just tried to gain some initial insights on how this effect could be, if there could be actually an effect or not. But
so how can you reduce the effect of shipping? Do you have any ideas how to recommend for these to
Well, for sure, first we would need to exactly have a clear idea of which are the species that are actually highly affected and the seasonality of those species and the distribution of those species. So for sure it would be we would need to drive first towards a reduction of the vessel shipping traffic. But maybe there are also some other limitation operation that we can employ for reduce the noise emitted by the vessels, of course. But first we need to have a clear idea of who is actually affected and how, and of course those devices utilizing this study can gain some insights, but maybe the best would be to employ D-tags, so actually tagging the animals and see their behavioral changes and gain more insights into what is the actual impact on.
You could avoid shipping in certain areas.
Exactly. Yeah, in this case, we've seen that this area could be actually really important for the feeding behavior of the species. So maybe there could be a other trajectory that that could be decided for the vessels themselves. So.
Thank you.
Thank you, Fallon. It's a very interesting, definitely relevant research. I just would like to elaborate a little bit further for the audience on why we should assess further the moon phase effect.
The moon phase. Yeah, this was actually an interesting part because I during my literature review, sorry, I'm just trying to get, during my literature review, I actually seen that it was already been studied the effect of moon phase on pilot whales, not actually long-fin pilot whales, but short-fin pilot whales. And it has been observed that during the full moon, there was a presence of longer and deeper dives. So I really wanted to include this factor in my study to see if I was getting the same result. But actually, I was expecting to have a positive relationship with full moon, and I got it with new moon. So, and looking at other literature, other studies have been like modeled the pilot whale's presence with moon phase, and again, they didn't get like super clear insights. But it is known that the light variation due to the moon phase can actually affect the migration of like animals below 1,000 meters. So I do feel that there might be like an effect, but it's probably a little bit hard to detect. And maybe again, like using D tags and actually tagging the animals, we can have like a finer and like more precise insights into how the animal is behaving during those different moon phases. But
Okay, and so do you think, you mentioned that the other species, there was maybe a correlation with full moon. You have an indication that the species that you address with the new moon, but other than the light effect, do you think the forces, the gravitational forces could be playing a role?
you mean like tidal? Actually, it is really hard to talk about like tidal effects in offshore waters because it's much more evident the effect like closer to the coast. So I believe that we could still like try to study the effect of specific like the tides offshore, but it's mainly a matter of like probably illumination fraction than like actually the force. But that's what I've gained from like literature and because
Okay, thank you very much, Akilita, for that very interesting presentation again. So now it's my pleasure to call into the stage Esmeralda Lopez Martinez, who's going to tell us about the disrupted harmony, investigating the effects of anthropogenic noise on humpback whale presence in the North Channel. Esme, you have 15 minutes, followed by 15 minutes of questions. What is yours?
Thank you. Thank you for the introduction. Well, welcome everyone. I'm Esmeralda Lopez and I'm going to present my thesis today, which was supervised by Dr. Joanna O'Brien from Atlantic Technological University in Ireland. So, first of all, cetaceans are highly vulnerable, with a third of evaluated species being classified as present on the IUCN Red List. They face many anthropogenic threats, and among these, anthropogenic noise stands out as a major concern. Irish and Scottish waters are very important for cetaceans because they host a wide variety of species. And among these, minke whales are one of the most commonly observed species, and humpback whales migrate to these regions. Then, to effectively monitor these animals is very challenging because they cover wide ranges and they're difficult to track. But passive acoustic monitoring provides many advantages. Among these, it's cost-effective, provides year-round data and across all types of weather, including hours of darkness. The acoustic data that was collected for this study was obtained from the Sea Monitor project, which focused on the seas around Ireland, Northern Ireland, and Scotland. And Atlantic Technological University was among the partners involved, and it was responsible for our marine mammals acoustic monitoring and soundscape. And this thesis specifically focuses on the anthropogenic noise impact on baleen whales, but specifically on humpback whales, conducting species distribution modeling. And even though minke whales were initially considered, the limited detected localizations prevented the further analysis with SEM. Regarding the study area, the acoustic equipment was deployed in the outer mouth of the North Channel, which is an understudied area. And the data collected was from stations 27 and 14, like we can observe in the map. And it covered deployment 1 that it started in October 2020 and ended in March 2021. The acoustic equipment that was employed were sound traps, which are broadband recorders, and this means that this allows them to record low frequency vocalizations from baleen whales. and they were set on a 1/3 duty cycle, so they were 20 minutes on and 40 minutes off. And then the data was stored in external hardware for further analysis. Both Minky whale vocalizations and humpback whale vocalizations were detected. For the Minky whales, the detection was manual, so it first required a downsampling with Tangbird and then a manual detection with Raven Pro. In total, 371 acoustic files were analyzed from September, October, November between 8 p.m. and 1 a.m. and this corresponds to the seasonal and dial patterns in the North Atlantic that are reported in the literature. And then for humpback whales, first pre-processing step was necessary, and then after this, all acoustic files from the deployment 1 were run through a coded acoustic detector, an inference mode to generate new annotations, and then the output generated was predictions per file, like a spreadsheet with prediction per file, and then from this, hourly presence absence datasets for each station and a global one were created. Then these are all the environmental covariates that were initially considered. Most of them have a daily resolution, except for the noise data, which has an hourly resolution. Then the primary productivity metrics have a 3-day lag, and all the values were taken as the average values within a 10-kilometer radius from each receiver's location. Then at the bottom of the slide, we can see the temporal covariates that were initially considered. And it is important to mention that the noise data was extracted from the first octave band, which is very similar to the frequency band that is used to assess the potential impact of noise on marine mammals. Then for the data exploration, the collinearity between variables was evaluated, and then after the collinear variables were removed, we came out with station-specific models and a global model. And for all models, 2 approaches were integrated in R. So one of them, this one is more manual and interpretative, and the other one is more automated. And the goal was to use generalized additive models or GAMs to explore like all combinations between individual predictors and two-way interaction terms, and then obtain the most parsimonious model with the lowest AS. Then moving on to the results for Minky whales across all 371 analyzed acoustic files, we only saw 2 faint calls in 1 file from November, but then we saw noise from potentially different sources in most of the files. So there we can see an example of shipping noise. And then as for humpback whales with SDM, this is a plot from the model with station 27. And we can see a negative nonlinear relationship between noise peak and predicted probability of presence. So then as noise peak levels increase, the presence of whales decrease. Then there we have interaction terms. So first we can see an interaction between noise peak and chlorophyll concentrations. So higher chlorophyll levels increase presence, but then even if in these food-rich environments, the negative effect of noise is still evident. Then here we have an interaction between eastward current velocity and salinity. So in less salty waters, whales prefer moderate currents, but then they avoid the stronger ones. Then under high salinity presence, remain relatively stable across all current velocities. And then here we can see that month 1 and month 11 were key months, so January and November, because we saw a negative relationship of presence with ocean conditions. More specifically, shallower mixed layer depths and weaker current velocities were associated with higher presence. And then these are the results from station 40. So again, we see a negative relationship between noise and presence of whales. And then there we can see the effect of month on presence. So we see that it was relatively stable, the presence across all months. And then there, again, we have interactions. So a combination of high sea surface height and high sea surface salinity decreased presence, which might indicate that whales avoid these conditions. Then there, current velocity also had a role, especially in low chlorophyll concentrations. So this might indicate that whales are more sensitive to changes in current velocity when food is scarce. And then there we see that increasing salinity led to decrease in presence, especially at high phytoplankton concentration levels. And then there again we see an interaction between chlorophyll and noise peak. So there we can see that especially when chlorophyll is low, so when there's less food availability, the negative impact of noise is stronger. And then this is the global model, which combines both stations. So again, we see a negative impact of noise on whale presence. And there again, we have month, like we saw in station 40. And again, it remains relatively stable across all of them, but then we see that in station 27, which is in darker purple, the presence was a little bit higher than in station 40. So this might indicate a presence for these sites. And then there we again have interactions between chlorophyll concentrations and noise peak. So it's the same patterns that we saw with the other models. And then there we see the presence peaked at intermediate values of sea surface heights under low phytoplankton concentration levels. And then for station 27, we have that the highest presence is at low to moderate current velocities under high phytoplankton concentrations. So then the main findings of the studies and the limitations are, well, for minke whales, the detections that we had in November contrast with autumn peaks in Scotland reported in the literature, but this discrepancy might have happened because of potential masking effects from shipping noise, for example, or also altered vocal behavior. And then for humpback whales, we saw a negative relationship between noise and presence, and this is consistent with known detrimental effects on humpback whales. And it is also known that noise can degrade or cause avoidance of suitable habitats. And then again, well, we saw a consistent interaction between chlorophyll and noise peak across all models. So this might indicate that whales might tolerate higher levels of noise when there's a lot of food, but then they prefer quieter habitats when food is scarce. And then at the beginning, maybe chlorophyll concentration doesn't appear as relevant because we were talking in a migratory context. But has been reported that there's opportunistic feeding during migrations. And also, well, including noise, foraging rates have been reduced with shipping noise and sonar. Then we found oceanographic conditions as key predictors, and this aligns with findings in the neighboring Melian shelf. Then our findings also align with preference for saline waters while feeding, but then avoidance of hypersaline waters. And also they employ currents that flow in their direction of travel, but then they avoid opposite currents. And we also found sea surface heights important in one of our models, and this supports sea surface heights as a potential important environmental driver. Then we also saw stable trends across all months, and this contrasts with reported patterns, migration patterns in the literature and peak sightings. But then also winter sightings have been reported in Scotland, and this can be explained by either the juveniles being present in the areas and they maybe require more frequent stops for feeding, or also non-migratory individuals or incomplete migrations also. And well, Caribbean and Cape Verde populations, they might migrate to these regions, but with different timing and routes. Then the main limitations are, well, first of all, that other modeling techniques might improve predictive performance, such as machine learning techniques or deep learning methods. Also, potential important predictors might have been excluded because we have static acoustic monitoring, so we couldn't include static variables like depth, distance to shore, roughness. And well, also we only included deployment 1 from Sea Monitor due to time constraints, but ideally we would include like also the other deployments. And well, also the ground truthing from the acoustic monitor, acoustic detector,
was not performed, so this is very recommended. But again, due to time constraints, we couldn't do it, even though the acoustic detector has proven to be very reliable for this area. And well, as a conclusion, we saw a significant impact of anthropogenic noise on humpback whales' presence in the North Channel, and this points out noise pollution as a critical threat for cetaceans, and also a need for mitigation strategies. And also, even though we didn't have a lot of minke whale detections, all the noise that we saw across the files might indicate that there's also potential negative impacts on them as well. And well, also our consistent presence, coupled with the opportunistic feeding, suggest that maybe the North Channel might offer foraging opportunities that are influenced by local hydrodynamics. It is also suggested to include human-related predictors such as fishing, dredging, chemical pollution, climate change, to understand the combined effects of this. And this is particularly relevant for the Cape Verde population, which is endangered. And well, our study addresses a gap in the migratory behavior understanding and also in understanding understudied areas, and contributes to the growing body of knowledge on this topic. Thank you very much for your time.
Thank you very much, Esmeralda, to manage to tell us such a nice but also complex project nicely on time, at least to us. Very nice confidence to explain your results, very interesting. Now I give the word to the jury, so Pele, would you like to start?
Yeah, I can start. How would you like to use your results from the study to improve the environment for the whales?
Well, I think this is important because the North Channel is an understudied area. So I think this as a baseline could inform further studies in the area.
Yeah. But you relate this to chlorophyll A. That's a simple measure of
the
proxy for food, I guess. Yeah. How can you use that? Because that's easy to record every day.
Yeah. Well, the thing is that because this is a migratory route, the North Channel, it wasn't considered as a foraging ground. So then to know that chlorophyll is very important there might also mean that humpback whales are more present, and then they not only use this area to migrate but also to feed and to do like other things. So then this increases the importance of the area.
But could you maybe reduce the ship traffic when you have high chlorophyll A? Should that help?
Yeah, maybe. Well, another limitation of the study is that they study the soundscape for the Sea Monitor project, but they don't have like specific sources of anthropogenic noise. So like they measure anthropogenic noise as a whole, but we don't know like which part belongs to shipping noise or to dredging or to other sources. So then it will be important to identify also like where the ships are, like how the traffic of the vessels is, to then like obtain more information about it.
Thank you. That's what
Thanks for your presentation. So, can you tell me your main hypothesis?
Well, the main hypothesis is that noise was going to be detrimental for both species that we studied, so for humpback whales and minke whales.
Okay, then how did you tackle this hypothesis? Which kind of, can you go back on your methodology you used to address, so to validate or invalidate this hypothesis? And do you think that your methodology is the most optimized for that?
Yes, well, sorry, I'll go back to methodology.
You can just talk to me about it.
Okay, there.
Okay.
Well, that was done with the generalized additive models. So noise was found to be significant in all the models. So I did 3 different models, like
What
one
do you
person.
mean, sorry, what do you mean the noise was found to be significant for
Because it was species distribution modeling, so then I included uh, environmental covariates and then also temporal covariates. And in the environmental covariates, I included noise as a potential predictor of whale presence. So then after I run all the models, noise was there. It was significant for all models. So then.
So, and do you think that this is new from the field, or this has been shown before with other methodology? And if yes, so what your model and what your study is important to perform, and what does it bring to the field?
No, this was done before many times with humpback whales specifically. So it is very known that anthropogenic noise is detrimental. But then what's interesting here is what I found with the interaction between environmental variables, like which other variables affect their, and especially in the North Channel, because it is known that the channel has a lot of diversity of species, but there's not a lot of details. So then there's still like obtaining more information. And also the soundscape was not really studied there. So from the Sea Monitor project, they found like they studied the levels of noise. So it's not a very noisy area. So then you would think at first that maybe it's a problem or it's an issue because it's not like a very like area with a lot of traffic apparently. But then with this study, we know that it is like it has an impact on that area, and also that there are other drivers like that are interacting between each other, like for example chlorophyll with noise. So
then you highlight a lot of data in your presentation. So for you today and to go next, so what would be the take-home message? The main message you really want to highlight from your study?
The main message, well, I will highlight like all the environmental variables that were important. Like for example, I found it interesting that sea surface height that predictor wasn't like very studied overall for humpback whales. So that was interesting. Also, the interaction between chlorophyll and noise that was present like in all models. So then that's also interesting because again the North Channel is just considered to be as a migratory route, but then it might also be a foraging route, and that's important. And yeah, I think that would be the main message.
Okay, thank you. So maybe one last one. So how can you use this data to help conservation?
Well, I think that it contributes to the general knowledge. It can be like used as part of the Sea Monitor outputs because they work with many species, but then in Ireland they work with marine mammals, and they already did some studies like with the data that they collected from Sea Monitor. So then this adds up to the database that they have, and then like it can be used as a whole to know more about the area, because one of the main goals was to obtain more information about these specific areas because they're understudied and they're not like we don't know what's going on there. So in that way, I think it will just give more information about it. And also they had studied harbor porpoises and dolphins mainly, and they haven't done a lot of work with baleen whales. So that's also important. They're also there, so it's also important to consider.
Thank you, Aldine. I think very important that last question of Aldine to highlight a take-home message that's aligned with the previous presentation, what we want to bring more awareness to the result. I just would like, Esme, if you can go now back to the end and to your conclusion slides. I think the one that shows points 3 and 4.
Yes.
Just to have a little bit more conversation around the subject. Remembering that we are streaming online and the recordings will be made available later, so I also hope that we can bring more awareness not only today but longer on with the outcome of your results. Yes.
Mhm.
So, do you know from the sea monitoring project to how extent, or if it's already planned to address this other varieties? For example, you mentioned chlorophyll and the relation. While you were mentioning, I'm immediately thinking, yeah, but what happens with that production in the times that we are living of intense dynamic climate change that affects all environmental variables?
Well, I'm not really sure if they're considering to consider these other variables, but there's a lot of data that is still there and that hasn't been used. So there's a lot to do with that. So.
What do you think about it, and what would you recommend in addition of next steps if you were already gonna have the power as an expert to make recommendations to scientists, to project developers, but also to the funding agents, as we know that we need funding to execute our projects.
Well, first of all, I think I will use all the deployments from CMonitor because it's a huge data set. So I only cover deployment once. I will cover all of it to see the seasonal trends. And then from the noise, I would also recommend for a CMonitor project for them to identify the sources. Because shipping noise is very important. It's one of the main sources of anthropogenic noise. But it's important to know to which extent it's contributing to the noise in the area. And then also to have more reliable environmental variables because, for example, for my environmental coverage, I use Copernicus and other platforms, but then there are other more local databases where more fine scale data can be obtained. So I think I will also recommend that to use more reliable data. That's it.
Okay, thank you very much, Ezra.
Thank you.
All right. So with this, we are concluding the first session in the whale room. I want to ask you all for a big round of applause to our three speakers of the day. Once again, Patricia, Margherita, and Esmeralda. And I wish you success and that I hope you can continue developing your research. I'm very pleased with the high level of quality of research that we have seen here today, and I hope that the audience online is appreciated or will appreciate as the recordings, as I just said, will be made available. So thank you again. Now we can already move to coffee break, although we'll officially start in about 4 minutes. And make sure to be a little bit before the starting on the room that you want to see after coffee break. See you later. Thank you.
Okay. Marisa, just to make sure, Marisa, you're here. Anna?
Here.
Here, okay, ready to go. And I think Niels is also in place. Yes. You want to come back to the first? It's your first slide? Okay. Just to make sure. So, welcome for this second session, just that you're sure, room 5, the well room. So, the jury member in this presentation will be Fabio Rindi from Marche, Université Polytechnique de Marche, and 2 jury members from Université Côte d'Azur, Hubert Bonnefond, and I will be also a jury member at the same time. So we will have a big change because Marisa is going, I think, to talk a bit about humpback whales. Yes. Anna about acoustic detection and description pattern of dolphins. So we're in the good room. And also Niels is going to talk about invasive algae and anti-inflammatory and antioxidant bioactive potential molecules, but we will see that. Okay, I remind you, you have 15 minutes sharp, and then we have questions. Oh my God. And I just have. So, Marisa, you have the floor.
Okay. Good morning, everyone. Did you know that diving behavior has appeared independently in different animal groups, as amphibians, reptiles, birds, or mammals? From these, 130 species are marine mammals, and inside this group, 94 are cetaceans, from which 17 are whales. All these animals dive to feed, but cetaceans spend most of their lives underwater, so most of their behaviors cannot be directly observed. For large cetaceans, as the baleen whales, there is also another problem of scale, and so control experiments cannot be performed on them, and we need other approaches to study them. Focusing on our largest species, humpback whales, as shown in this figure from the Blue Corridors report in orange, are distributed worldwide. And so, modeling becomes an interesting approach to study them. Considering that, I am Marisa González Felix, and I'm going to try to fathom about a mechanistic model to conquer diving limits for humpback whales. To start, I want to show a general representation of a dive. First, humpback whales are central place foragers. That means that they are constrained to return to the surface. Second, their dives are continuous. That means that the individual moves in space and time in a cycle that is repeated. With this, we aim to design a model to study the diving behavior of humpback whales. We focus on the individual and use a mechanistic approach that relies on the physiological and the mechanical processes to simulate a dive. For our model, we consider that to generate a movement, a force is produced, and this force is linked with an energy and oxygen consumption. This approach contrasts with the usual types of models that has been used as the optimal dive strategy that relies on population-based estimations and frames the dive as optimal times and depths to use its stored oxygen. All this constrains its ability to study dive limits, as is shown in Tyson 2816, where they try to plot the foraging time against the maximum dive depth, and the estimated trends, as black lines, do not match the observations in gray that are done at the individual level. So the optimal strategy shows movement as ranges of time and depth, but in our model, we put a sum of forces and generate a 3D trajectory in time and space. This is the type of measurements usually seen for biologgers placed on individual animals and allow us to explore different dive scenarios with changes in buoyancy, dive depths, or feeding events. From the physiological processes, the optimal strategy
strategies relies on allometry relationships globally, even for their energetics, and these are usually estimated from other species. But in our model, we use the oxygen and energetic metabolism based on the description of actual physiological processes behind them. So we have a better grasp of the diving limits, and we can try to identify the oxygen limits on the dive. And then the optimal dive strategy uses statistical trends found in the population to study the diving behavior of a species. But the mechanistic model, we will focus on the individual, and so it allows us to use the biologger data directly and develop a dive ethogram able to analyze ecophysiological properties of observed trajectories. With these objectives, we designed a model. The first step was to describe the whale. The whale was represented comparing its mass with different lengths, surfaces, and volumes, including the volumes for the oxygen metabolism compartments, lung, blood, and muscles, and the capacities of engulfment of water while feeding. All these relationships are based on an assumption of isometric growth. This means that the individual does not change shapes while growing, as it can be observed in this figure with a mother and a calf. Then, how is the movement of the whale model? We consider movement as a set of interactions between the main forces, buoyancy, weight, drag, and force produced, and its resultant accelerations. And then the biologger records the position of the whale in space, and this has to be converted to the geographical axis. Also, the whale produces a force to move by flocking. This is derived in the model as a propeller force that shows the main characteristics of an intermittent propulsion. This is observed in data, for example, of Goldbogen 2012, where we see intermittent peaks of flocking in time. Next, we have to link the movement with the metabolic energy. This is done deriving from an expression from the study of Heath and Garney, 1997, where the active metabolic rate in joules is a function of the force to the power of 3/2. That is an isometric property. And now, we describe the oxygen metabolism by 3 main compartments: lung, blood, and muscles. Lungs get oxygen from the air and transfer it to the blood. In the blood, the oxygen can be transferred to the organs as the basal metabolic rate, that is the minimum energy required to maintain the organs in the body. Or it can be transferred to the muscles, where the oxygen is supplied to the active metabolic rate, that is the energy required to produce movement. However, this model is adapted from the commonly used human respiration model of Popel 1989. That is a free-diving human model which still has many uncertainties. And all of this shows the limited understanding we have of whale and diving physiology. Also, some clarifications. Starting with the atypical mammalian circulatory system, the model divides a compartment of blood from oxygenated blood that goes to the tissues and deoxygenated blood that returns to the lungs and heart. We will only focus on the oxygenated blood. And from this, we consider that the oxygen processes are fast processes. And so, this showing the test model simulations for the surface conditions, where the breathing rate fluctuates in time according to the effort produced, but the transfer rates are fast enough to maintain the oxygen storages at equilibrium. The next step was to develop the diphethogram. I will not show the diphethogram itself, but we will point out some characteristics. First, they are divided the dives are divided into different behaviors that are body position based. Then, short time intervals are required between the behaviors to change body position. And finally, even if this is still a preliminary study, it allows us to start studying the limits and signals for these dives, and also add probabilistic decisions in future studies, as the dashed line shows what an interrupted dive could look like. Having a functional model, we built the different scenarios from our literature review. We consider a range of sizes from 4 to 14 meter length, but we will only show results for a 14 meter long whale. Then all simulations are complete dive cycles or surface to surface. And I will discuss part of our results about the blood oxygen limits, the feeding events, and the dive ethogram of an actual dive. The first scenario shows 50 and 10% blood oxygen threshold simulations. This is a test of the relative quantity of oxygen left in the blood to return to the surface. In the graphs, we can see the liters of oxygen stored in time for the 3 compartments: blood, lungs, and muscles. The dive starts when the oxygen storages deviate from equilibrium. For the 50% case, at about 6 minutes, the individual reaches the surface and recovers its oxygen. But on the 10% case, the dive becomes anaerobic, not having enough oxygen on the return. So there may be a limit of oxygen during the dives, but other possible signals, as the nitrogen and CO2 concentrations, can be an option, or the signals could be surpassed as it has been observed in humans. Also, the model overestimates the energy consumed, since some dives do not have enough oxygen to complete the dive, even at depths that are usually seen in literature. This could be produced because of the use of allometry-based energetics, and so this highlights the need of physiological measurements for whales. Next, I want to show the feeding behavior for individuals on 50-meter dive simulations, one without feeding and the other with one event. The graphs show the energy rate in liters per minute in time as the black line. The red line is the mean energetic consumption. And the gray area on the right delimits a highly increased energetic consumption that is the feeding event. The feeding event is divided into 3 main phases: engulfment preparation, where the main peak of energy is found, the engulfment of prey and water, and then the filtering. But still, this model overestimates the energy consumed. So this limits the depth of the dives that we can model successful simulations. And also, researchers are studying feeding mechanism models, but filtering is still not well known, and these models are still too complex to implement here. Finally, I want to show you a comparison for a feeding dive. In here, we see the estimated pseudo track from the biologger, and then the model trajectory with the same rotation data. The figures show the 3D position in space in meters, and the black and blue lines are the different body postures or behaviors. The pink bands shows the time intervals between to change the body posture, and finally the gray is the start of the dive. That is the same for both. The estimated pseudo track shows the first classification ever done for observed dives. And then the model trajectory shows that our model is able to simulate realistic trajectories. All this highlights that the dive ethogram is a step forward on the knowledge of dive limits and behaviors. And also, this is necessary to automate classification of biologger data archives. So what did we learn in general from these scenarios? The energy consumption is overestimated in the model, but highlights the need of measurements for whales. Then, feeding generates a high energetic consumption on a limited time. And then that this dipethogram allows the first classification for observed dives, and also that model dives can simulate realistic trajectories. So in this project, I have been discussing the capacities of the optimal strategy to simulate and study dive limits, and have used a mechanistic approach to study them. From this, we have found that still physiological measurements are needed for large marine mammals, and that our mechanistic model is compatible with the data gathered from recent biological studies. But what is the most important outcome of this project? For me, is the collaboration initiated between field scientists and modelers. With this figure, we can see that groups from California, Oregon State, New Hampshire, and Iceland universities have provided us with data and information to improve our project. And I thank them all for their support. This brings new perspectives to advance on integrative approaches to whale behavioral studies. Again, I am Marisa González Felix, and this is the end of my presentation for my master thesis. I am now ready for any questions you might have.
Also, thank you for the timing. So we can open the floor to questions. We're also ready to welcome any questions potentially from a supervisor, maybe, or I don't know. So, you want to start?
Okay, very nice presentation. I enjoyed it very much, and it seems very interesting, even though I have to say that this is a field which is totally out of my personal expertise, but still, I like it very much in the whales. I wonder, okay, the type of logging data that you use, are these publicly available? Where did you get them?
I will tell you, well, as you've seen, we've collaborated with different groups, and so this data right now that is showing here in the estimated pseudo track is from a public data that is the Goldbogen Data Laboratory. And from here is one of the latest research from humpback whales. So this is publicly available, but from other researchers, they've also shared with us some information and some data that they are from their own studies and that they are private.
Look, I'm not sure if it is you are the right person to ask this question. But how do these people actually manage to grab a whale and put a lot of
No, yeah, I know how to do it because I had to study a bit of the biologists. So the thing is, well, they first try to be as less impactful as possible to not disrupt the behavior of the whales too much. So they usually go on small boats, and then they have a large pool of, I think, 4 to 6 meters. And at the end of the bowl, they put a small biologger that, well, I think I can go down and it's, here. So it's, I think it's almost more big here than usual. But they put a small biologger at the end, and so they approach the whales around 4 to 6 meter distance and put it here, but this is also depending on how the whale is, so it can be positioned here or more near to the tail. But
you say that it's really easy to do, like
you just have to be a bit like not shaking to put the ball in the whale. But that's it.
Normally, once the logger is fixed, how long it will remain on the
okay. Yeah, well, it also depends on the biologger, but because also depends on the capacity they have to record. But well, first the biologger can move, so they can put the biologger here, and with the movement of the whale can be a bit reattached and go and move a bit. but this can also be measured for them. I didn't do it itself, but this can be
Okay,
measured.
I mean, then this data that they obtain from the loggers for how long?
Yeah. And then for how long? There's some that was as short as 4 hours, 6, but there's others that can last to 24 to 48, and I think some can go almost to a week, but that part I'm not sure about the 24 to 48 days. For high precision DTAC data.
So then one last thing, look, I know that the humpback whales actually have the very long, large scale migrations. They move from tropical waters to polar waters. I wonder if the models that you prepared, okay, might be different depending on the latitudes of the geographical areas.
I understand that question. Well, right now, as I said, this is a preliminary model. It's mostly based on the individual itself. So indeed, we had some problems because, as I've been repeated through all the presentations, there's not that much of physiological measurements, so the model is a bit difficult to really understand on that part. And so our first thing was to, apart from doing all these individual, to put some environmental cues as the temperature, the latitude, the depths, the salinities, but this is not implemented in the model. However, it's a step forward to it, and also because this model is, it has the idea from the model first presented from my supervisor, that is a global migration model for humpback whales. So yeah, it was a bit linked with this.
Thank you.
Thank you very much.
Thank you.
It's not very clear for me, how did you calibrate, or if you calibrate your model, and if not, why, and how you can do that, and if yes, can you explain it a bit more?
Okay.
Slide question.
Well, I don't know if I have any slide where I say, so I'm going to talk, and if there's any of that. So calibration of the model. Right now, the most calibration of the model was first to find the parameters as reliable for the humpback whales. So that was one way to calibrate or try to make better the model. However, apart from that, as we said, there's some still paths that are not well known. And for future improvement of the model, it would be nice to do, for example, the analysis of the sensitivity of these parameters, because this is not done in our model. And for example, try maybe with different parameters and better comparison with the data to see where we can improve and implement better this pitch, yaw, and roll that are the different rotations from the city. I don't know if more.
So, for me, a calibration is you take real data and you put your model in front of and you see if it's matched. You have to check to estimate different parameters. Did you do that?
The data we have is from these people, and it's DTAC data. So we have the depths, we have the pitch, yaw, and roll, and then we have estimated from that, the rotation itself, because they have to do the changes, and then the pseudo track, the velocities, and the accelerations. So we did not, in this version, compare directly and statistically with these models, so we cannot say that they are significantly this percentage similar with the data. And so that's for that part. And then for the parameters, we took the parameters from recent previous literature that was working on humpback whales. And one thing to improve or to try to get together and have a better measurement of them was actually what I said here, that is the isometric relationships or the isometric growth. So considering that the whale has an isometric growth also allow us to compare with previous literature and have a better link between all these parameters.
Okay. So yeah, there is still works concerning calibration. You still have job to do concerning calibration because the calibration.
Well, yeah, you guys, well, I've just seen before, I don't know if I can go back to that. We see a more or less good realistic trajectory. It's true that in some places there's still some calibration and some behaviors that are not exactly represented. But indeed, being that a preliminary study and a first try to do that, we are happy that, well, that the model indeed could represent these trajectories, at least on a general view. But yeah, of course, we need to improve it, to continue comparing with data, and include more information on it.
Time for one more question?
Yes.
And just how can you give us to be a little bit critic concerning you use some human model and you apply it to wells, if I understand well.
I used a mechanistic model to apply it to diving behavior of whales. Yeah.
Mechanistic coming from some human behavior, if I not,
okay, that part.
How can you? What is your point of view? Because humans are not a big, very good diver, and whales a little bit more. So probably there is some difference explaining that. So what is your point of view?
Yeah. Again, I think it was around here, so we come to this. In these parts, because it's true that for the mechanic part, there's more information and more studies for the mechanicals and how they move. But for the oxygen, it's what they say, we found a real problem of that there's no physiological measurements that we could apply to have this type of model of really with the processes from humpback whales. So we tried to start comparing with how it has been studied, with the theoretical work that has been done for the oxygen metabolism in whales. And from this theory, we found that the most common model used, even for marine mammals, was this from human respiration model. So yeah, that arises, and in my opinion, again, this need of really knowing what's happening in the physiology of whales and how maybe blood measurements with new biologists that, for example, can take the temperature and also I think the oxygen, the breathing at the surface. So all this could improve this vision we have, and I think my opinion is that we need them.
Yeah, thank you. It's indeed important to, when we develop models which are complex, to always be clear with the limitations of the physiology, and so that's a paper from the 80s. But of course, it's not like we can measure the blood measurements in whales yet. Well, you talked about the fact that you actually, for size, I would like to know if you know how size impacts your model. You say it's kind of linear with the effect of the size on your model?
No.
But my question is that I guess, I don't know if you consider that babies are not maybe feeding on the same food and have not the same metabolism, so than an adult of 14 meters, so how do you consider that?
Okay, how did I put it? Indeed, we didn't specify more than the weight of these surfaces, these volumes, or these masses for describing the whale. So we did not a specific differentiation between, for example, a calf from its physiology and an adult whale from, again, this feeding and physiology.
You haven't done that in your model?
Yeah.
But I know that, do you know if for real you have a difference?
Well, if on the diving behavior,
or on the metabolism generally of babies, because they don't eat the same things.
On the metabolism, I'm not as sure, because same, the physiology, they say more or less how they feed and those. But in some studies, I have found that the diving behavior of calves and mothers indeed can be very similar.
Okay.
So you can see a 4-meter whale to dive 100 meters and come back to the surface at 10-15 minutes. That is more or less what an adult does. Following. So yeah, I think it's controversial, meaning like it needs more study, and it could be interesting to really know the differences between them.
We have maybe a short question, if you can try with a short answer. It's not a short question, so how is temperature really part of your model? Can you tell us how it impacts the results? I know if you've done so, no sensitivity analysis yet.
No, we didn't do the sensitivity analysis yet.
But do you know how the temperature impacts your model? And my second question behind that is, generally speaking, how can we use your model in a climate change context?
Okay, well.
Not only temperature, also.
Well, as one part, temperature for the model metabolism of the whale itself, as we know, whales and all animals have indeed some processes, some behaviors and some mechanism to make less big the effect that the different temperatures can have. However, we also know that in very high changes on temperatures, this, could be it could change the behavior of the whale. However, I think also temperature has a bigger effect apart from the whale itself, that this is one of the climate change effects or points, and so this can also change its food itself. So changing its food, that would mean that maybe the movement and migration of the whales can also change, and they may spend more energy, depending on how its food changes, on doing these migrations, this movement, this diving energy. Interesting for this model is to next implement this energetic gain and these food particles to know how is the balance between energetic consumption and gain. And, I lose myself with that, but from that this. And the other thing, how it could be applied to conservation, you mean?
No, I think you answered globally on the question, yes, with climate change in general.
Okay, so fine.
Unfortunately, it's always over. It's really interesting, so we could have continued, but I think you want to finish it and upload?
Yes.
So congratulations, Liza. So we can continue with you now.
Good morning all, my name is Anna Sam and I'm here to present my project about seasonality of acoustic detections and distribution patterns of dolphins on the northwest coast of Portugal. About introductions, I think we all know that marine mammals are vocal animals, that they rely on sounds for mostly everything. So it will be interesting to use the acoustic techniques to study them. One of the acoustic techniques is passive acoustic monitoring, which is a non-intrusive technique that, as I said, we can use for studying cetaceans. It has some advantages compared to the traditional disruption rates, and is that it does not depend on the sea state and the weather conditions. It can be used for long-term monitoring. It gives us information about the animal behavior, but it also can be used to study possible impacts caused by humans. And lastly, it allows to work with species that are harder to observe. This technique also allows us to study ambient noise or background noise, which is defined as the noise associated with the background noise emanating from a myriad of unidentified sources. It's always present, and it was originally formed by natural sources, like natural events such as wind or waves, for example. The marine mammals developed their acoustic and vocal specializations in historical levels of background noise, which is the problem. With the growth of industrialization, these levels of background noise has been disturbed with an increase of sound from human sources. The use of palm in the northwest coast of mainland Portugal is one of the less studied marine mammal topics. Basically, there is a gap in the acoustic studies of cetaceans. That's where the Atlantic project carried by CIMAR is trying, one of the objectives is trying to reduce this gap. This project is, they are trying to collect different types of data to create a platform for monitoring and sustainable exploitation of the North Atlantic Ocean and its resources. In the Portuguese waters of the Iberian Peninsula, we have 3 species of Delphinidae family that are regularly observed. These ones are common dolphins, bottlenose dolphins, and resource dolphins. About the objectives and hypotheses, The main objective of our study is to see if there are temporal or spatial patterns in the distribution of vocalization of dolphins in the area. And then we have 5 particular objectives. The first one is determining the distribution of dolphin vocalizations present a temporal or spatial pattern, and it is influenced by the background noise and the presence of snappiness rings. Second is to see if the distribution patterns of vocalization vary between the classification of clicks and whistles. The third is to see if there is an impact of the background noise on the acoustic detections of overall and for clicks and whistles separately. Fourth is to compare the visual data with the production of clicks and whistles. And lastly is to see if there is a correlation between the values of background noise of the area and the presence of human infrastructures in the area. About the hypothesis, we have 3. The first one is that there is a non-homogeneous distribution pattern of dolphins, both spatially and temporally. Second is that there is a correlation between the highest values of background noise and the human infrastructures. And third is that there is a negative correlation between noise and dolphin presence. About the methods, the study area is in the northwest coast of Marina, Portugal, between the towns of Framina and Espinho, and both coastal and offshore regions were covered in the study. For the data collection about survey design, the study was inside of the framework of the Atlantida project. It was started in summer 2021 and finished in winter 2024. About 2 seasonal campaigns were carried per year, and the design was 8 parallel transects that they were perpendicular to the coast. And per transect we have 4 sampling stations. The visual survey, always a minimum of 2 persons were doing the survey on top of the boat. When a sighting was done, we need to write down the species observed, the group size, the behavior, the presence of calves, and the GPS location on the angle and distance from the boat. For the acoustic sampling, we use an IRC Listen HF hydrophone probe, this one here, and we program it with the Market Ocean software to record at a maximum waveform of 100 kHz. The sampling was always the same. When we were in the sampling site, the engines of the boat were turned off. We did the deployment of the hydrophone and we left it to drift for approximately 10 minutes. For the acoustic analysis, all the analysis of the audio files were carried in Audacity software, where we obtained the spectrograms. These spectrograms were visually analyzed by a human observer, basically me. We searched for biotic sound sources. These biotic sound sources we are referring to dolphin whistles and clicks, but also snaps of snappiness rings. And then we classify them in 3 categories. These categories are category 0, there's no biotic noise. Category 1, there is presence of biotic noise, but it occurs occasionally. And in the case of category 2 is a high density of biotic noise, and that has a high frequency of occurrence. It's important that everyone knows that it was not possible to distinguish between the 3 species of dolphins I spoke before, only based on the vocalizations. For the analysis of the background noise, we select the most silent minute of the entire audio file. This minute was analyzed in Air Studio using the Pangai package that was developed by Nelson Lecole in 2015 in this paper here in testing. And that code was measured in decibels. About the anthropogenic sources, we identify the main anthropogenic infrastructures in the area. We download the data from EMODNet, and we use ArcGIS Pro to obtain spatially explicit covariates. These explicit covariates are for each sampling station, the distance to the 2 main harbors in the area, which is Lesois Port and Bionario do Castelo Port, and the distance to the wind farm, and the root vessel density and fishing vessel density for each sampling station. For the statistical analysis, all the analyses were carried in our software. We based the model selection in a Kaika information criterion, and we established the threshold statistical significance is 0.05. We carry multiple generalized additive mixed models in which we include the sampling site as a random effect. As I said, I carry multiple GANs, so we have different response variables. We have binomial, that it was presence absence of acoustic detections, clicks and whistles, and a continuous variables. Also for another analysis, we carried an ANOVA with a Tukis test. For the results and discussions, for the study of the distribution patterns of vocalizing dolphins, we carried all these models. The one in red is the best fitting model. This model was able to explain a 37.7% of the total variability in the data. And as we can see in the outcome of the table, season had a significant effect in the detection of vocalizing dolphins. This effect is basically that we have a higher detections of dolphins during summer compared to winter. In this graph, that is the prediction of the model we work, I explained before, we can see a different distribution pattern between summer and winter, having higher values like detections during summer. Similar results were found in other studies in which they explained this difference in changes in the sea surface temperature because of the migration of the main prey, or because basically they are searching for better conditions for calving. For the study of the distribution patterns based on the dolphins' productions of whistles and clicks, in the case of whistles, again, all these models were carried. The one in red is the best fitting model, and it was able to explain a 33.4% of the total variability found in the data. As you can see in the outcome of the table, season and the interaction between latitude and longitude for summer, they were significant in the production of whistles, having a higher detection of whistles during summer compared to winter. In the prediction of the model, we can see that there is a different distribution pattern between summer and winter, with higher detection of whistles during summer. Multiple studies have similar results, and they explain this because they have a higher social interaction, or because they are periods of reproduction and calving, or basically because you use it as an individual recognition, the whistles. In the case of the production of clicks, all these models were fit. The one in red, again, was the best fitting model. This model was able to explain a 16.2% of the total variability found, and the presence of happiness ring had a significant influence in the production of clicks. This significant, basically, this influence basically is that when the happiness ring were present, the production of clicks decreased significantly, and we could not see a difference between the distribution pattern of summer and winter. But about the results, other studies have similar results in which the snapping rings have an influence. This influence can be that they are generating an auditory masking. With this, I mean that they are interfering in the vocalization, in the production of clicks. While in other studies, they found that the tried dolphins stop echolocating, and they change the swimming direction when the snapping rings are present. Okay, so for the study of the difference of the background noise on acoustic detection of dolphins and the production of clicks and whistles, for the model selected for the three of them for the acoustic detections was the model selected for the acoustic detection was able to explain a 21.9% of the total variability. For the model selected for the production of whistles was able to explain a 16.4% of the total variability in the data, and for the production of clicks, it was able to explain a 14.91% of the total variability. The outcome of the three models that we can see in the tables is that the background noise didn't have a significant influence in the production of the detection of acoustic detectors and the production of whistles and clicks. But in the three models, the site index, that was the sampling site I was speaking before, the methods, had a significant effect. This means that the acoustic detections and the production of whistles and clicks is changing significantly across the sampling sites. This may be suggesting that these three models may be suggesting that the animals are getting used to the background noise. Similar results were found in acoustic detections in which they explain that the capacity the animals have, the dolphins in this case, have to adapt their hearing sensitivity to the ambient noise. This is called auditory gain. In the case of whistles, it will be interesting to study in the future the whistles' characteristics, because we found studies in which they present compensation strategy in which they make shorter whistles when the higher the background noise values are. And in the case of clicks, we found similar results in which the animals had gotten used to the background noise, but also different results in which the production of clicks decreased when the background noise increased. For the comparison between the classification of whistles and clicks and the visual data, both ANOVAs for whistles and clicks were significant. And in the case of the whistles, the results showed that the pairwise comparison between the whistle category 2 and the whistle category 0 was significant. This means that the difference in the mean encounter rate was significant between the two categories, being higher for the whistle category 2. And in the case of the test results for clicks, the pairwise comparison between category 2 and category 0 was significant, as well as the pairwise comparison between click category 2 and click category 1. This means that we have higher encounter rates in the clicks category classified as 2. This outcome can be, these results may be suggesting that we record the animals while they were feeding and calling, traveling, because this is the most common behavior, and they use both clicks and whistles for both of them. In the case of the study of background noise correlation with human activities, again, these three models were carried, and the red one was the facility model. This model was able to explain a 19.19% of the total variability in the data. And as we can see in the outcome of the table, season and the root person density had a significant effect in the background noise values of the area. In the case of the root vessel density, we can see that there is an increase of the background noise values the higher the density of the root vessel density goes. This makes sense because the seeping noise is one of the biggest contributors at a global level of ambient noise in low frequencies. In the case of the season, we can see that winter has a significant higher values of background noise compared to the summer. Also, we can see here the difference between summer and winter. The blue dots are winter and the red dots are summer per sampling station. They are higher values, and we have other studies have similar results in which they associate these higher values of winter due to change in the propagation conditions of water and the presence of wind. On the other hand, in Via Formosa, they found completely the opposite, in which in summer they have the higher values of background noise due to an increase in the boat traffic. And lastly, the conclusions. We can conclude that the vocalization of dolphins present a temporal pattern with higher detections of summer. In the case of the detection of dolphin whistles, they present seasonality, again with higher detections of summer. In the case of the detection of clicks vocalizations, they are influenced by the presence of snapping streams, decreasing the clicks when the streams were present. Neither the vocalization patterns nor the whistles and clicks were influenced by the background noise, and there were higher detection of clicks and whistles when we have higher detection of clicks and whistles when the background when they were sighted in the area. And lastly, the only human source that contributed to the background noise was the density of seeping roots. But we also found that the background noise in the area had seasonality with higher values in winter. Lastly, I would like to thank the people from CIMAR because they provide me with the data, and my three supervisors, Thiago Marquez, Ana Bio, and Mafalda Correa, for the continuous help. Thank you.
Congratulations, Anna.
Thank you.
Here we go with you, Hubert, first.
Thank you very much. You use different kind of model with different parameters to explain
The distribution patterns mainly.
Do you have some biological explanation? Because you have a list of models, you choose one based on statistic. Okay, it works statistically, but maybe it's correlated with the number of cars in Washington, and it's not a good model. So how do you explain why this one and not the first one? Do you have some explanation, biological explanation?
Why I choose this one instead of the other ones? Being honest, I only focus on the IAC values for the selection of the models. We choose these ones because we want there is no study the response variables that we choose them because in the case of the season and the interaction between latitude and longitude, there is no many studies in the northwest coast of Portugal about that, so that's why we choose them. And in the case of background noise, we wanted to make an impact assessment to see if they were an impact. And the person of snapping streams, they make a preliminary study in which they see that they were some influence in the detection of dolphins. But basically, to choose the model, I only based on the IAC model selection.
Okay. Another small question is you said you can't distinguish with the sound the dolphin species. Do you have an explanation? Do you think it's because they do the same noise, or do you think there is another explanation?
It's because there are studies that they were able to discriminate between the two, between different species of dolphins, but the thing is that you need to have automatically identification. The name, but there's a way that it was automatically you have some code and you can include the audio file and it will be able to discriminate between them. But in the team, this is a really basic, it's the basic line of the studies they are doing. There's a PhD girl that is going to work with that in the future, so they give me the basic line, so I was not able. I know that there are studies that they have managed, but in my case, we only do visual analysis. So that's why we couldn't differentiate, because visually, or not even listening to them, we're able to distinguish between the three species because they are quite similar.
So to do that, you need more of you need a computer analysis or
you need
to analyze the signal.
Yeah, in the office I was working at, there was a girl that was doing that with other species. First, you need to know Python. I don't know Python, and they told me like, yeah, you will need time to do that, and because of the time, I was not able. But it would be interesting. But yeah, it was. You need like more knowledge about that.
Thank you once again. This was very interesting, but it's not my field. Not that I know something there, I was wondering, yeah, but I love dolphins as well. No, look at since in your model, the latitude and longitude that they came out as significant. I was wondering if this species of dolphins in terms of geographical distribution, do they have a wide geographical span?
Yeah, like the distribution range of the animals.
Yes.
Yeah. Well, it's just to have the picture, the cute picture of the dolphins. I'm sorry. Okay, so the three of them, they have like a general distribution, like they are quite present everywhere. Mostly the common dolphins and bottlenose dolphins are in temperate. In the case of tropical dolphins, they are everywhere, but in temperate and tropical waters. In the case of common dolphins, they are in warm waters. But this guy here is cosmopolitan. The thing is that it's one of the less common dolphins that you can see of the smaller delphinids. Like it's one of the three is the less common to see in Portugal, at least.
I was wondering if maybe from this point of view there might be some differences in behavior where different latitudes or in different regions.
Yeah, for the case of the bottlenose dolphin, I know that there is a study in Panama, in the Canal of Panama, that they found like a spatial association that was significant, and it was due because the noise their main prey was making. So they changed the spatial association in depending on the main prey. So yeah, I was like there's different behaviors for sure.
Okay, and another thing which is not directly related to this, but I mean since the noise produced by the human sources, often would be also associated the light pollution. I was wondering, is there any information about the possible effects? No, sorry, I realized
No worries.
out of the scope.
There is some studies. I don't know. I haven't found any studies about light pollution because I was focused on the noise pollution. But in my case, I don't think you will find anything because all the samples were done during the day and it was only 10 minutes. So it would be difficult. But I know that, for example, other studies have done like dial cycles, like 24-hour records. So maybe in those ones you can find something. But the ones I have seen with the total like 24-hour cycles is usually for changing patterns of the vocalization of the dolphins. They are not focusing on the like illuminic pollution. Yeah, thank you.
Thanks.
Okay, maybe coming back especially, so can you come back on one of the maps, longitude, latitudes?
The models?
Or maybe this one, yes.
Okay.
First. What's the distance, again, of
From
your area of study? What's the size?
I don't quite know, but the distance between the transects is 6 nautical miles.
You're gonna have to help me then.
Okay.
No, but roughly, are we talking about 10 kilometers? I don't think so. 100, 1,000 kilometers?
You're catching me there.
My question was, I'm not sure I'm going to help you with my question then.
Okay.
But if you are looking at longitudinally, okay, east and west, have you checked if it's, so the difference you could observe especially is linked to the difference in longitude, or it's actually kind of the same longitude, and it could be also the distance to the coast? Have you checked that, if the distance to the coast has an effect?
We didn't, like, I always work with the interactions between season. Well,
Take your time.
yeah, no, it's because I'm trying to remember because the first basic models I did before these ones, I was working with the coordinates separately. And if I'm not wrong, longitude was significant.
But do you think it will be longitude?
For-
Given the
because of
fact
the
that you have a
distance to-
really long, short span of longitude.
Yeah.
It's not like a difference between, the east and the west of the Atlantic
Now
Ocean.
that you're bringing out, I remember that that one was significant in the. But then we wanted to know if there was like an interaction between the coordinates, so that's why I removed the model and put the interaction. So, but yeah, it can be distance to the coast.
And do you know if you have any difference in temperature latitudinally? North south.
I don't know, but I know that while we were doing the samplings, they were taking measures of temperature also, but they didn't give me that info. Yeah, no, they didn't give me that info, but
No,
because
I heard the question also with that you were on the boat, so you were a human.
Yeah.
Okay. Have you experienced some difficulties to spot the dolphins visually as you were getting away from the coast, for example? Or because
so
I guess
we have like problems watching like seeing dolphins when more the farther away from.
yeah.
It depends on the species. For example, in my experience, I only went to 2 days of sampling.
Okay.
When we were leaving the port of when we were leaving the mouth of the Dover River, this year, I was able to watch bottlenose dolphins. But for example, the more farther away we went to the coast, we were only seeing common dolphins. In my experience, I don't know if it's the same all the time when they have gone because there's a few seasonal campaigns that they were before me. I don't know in those cases, but in my case, I see that more common dolphins are here than here.
Okay.
But yeah, being honest, it was really quite difficult because the sea state that day was horrible. We were dancing in the boat, so it was quite difficult.
So it could impact also the observation.
Yeah, for sure.
You have another question? Not yet.
No.
Oh, we.
Ryan, maybe one more question. When you analyze the noise, you choose a very specific sequence. You choose the sequence. The 1 minute was the noise.
The background noise. Sorry. Yeah, I just got it. Sorry.
when you analyze the noise.
Yeah.
Your sequence, it's a 10-minute sequence, and you choose just 1 minute, and it seems the minutes where the noise is lower. It's what you explained. So why this change?
So the intention was, it was the most silent minute that we try not to have biotic and noise, non-human noise. So we have, so it will not be biased because if we have a boat crossing by you will have a higher detection of background noise, but it's not the real.
Okay.
So, we were trying not to that it was not biased by that. So we tried to have the minimum we could get. Sometimes it was really difficult because the boat was under 10 minutes, so it was hard, but that was the intention.
I have a question you already answered. I think I was taking note of this answer.
Okay.
So, you said at some point that the animals may get used to background noise.
Yeah.
Can you just remind me why?
So it's just a suggestion. It's not that the model is saying that. Is that maybe the background noise did not influence that because they can get used. We don't know. Like maybe we need more studies to get that. But it's true that some papers have found that in some cases, not all of them, they have the same level of detections, and they explain this because of some dolphins have an auditory gain. That is, they can adapt their hearing sensitivity to the ambient noise.
On paper, on literature, but what is it on your results that shows that could? No, it was just based on literature.
It was based on literature. Yeah, no, it was just a suggestion based on literature.
Okay, I think we're all good. Congratulations. Let's go for Niels. Not sure we're going to talk about whales and dolphins now.
No. Well, that's why you're here.
That's why you're here. Okay, everyone, you still good? getting warmer and warmer with the day. I have to tell you.
All right.
All right.
Everyone's ready. Welcome everyone. Today I'm going to talk about my presentation about bioactivities of two invasive macroalgae species, Rugulopteryx okameri and Asparagopsis armata, and their applications in chronic inflammatory diseases. So let me get started with a quick introduction about chronic diseases. The prevalence of chronic diseases is rising at the moment, and to give you an idea, 74% of all deaths worldwide is due to chronic diseases, which is about 41 million annually. These diseases include a lot. It can be cardiovascular diseases, osteoarthritis, also cancer and diabetes, and all of them together, they really pose a threat to healthcare systems worldwide. And so this rise is due to several factors, including metabolic risk factors. It can be sedentary lifestyles and unhealthy diets, but also due to environmental pollution, such as air pollutions. And then the life expectancy is continuously increasing, so the demographic shift is expected to worsen this trend, as chronic diseases are also age-related diseases. There have been a lot of advancements concerning therapeutics, but they're often limited in efficiency, efficacy, and also not always selective, and they might have potential adverse side effects. The treatment can be long and expensive, so there's a cost associated to it, and furthermore, the mechanisms driving inflammations, which is a key factor in chronic diseases, is not fully understood. So, non-steroidal anti-inflammatory drugs are now used to manage chronic diseases, and the increase has also used over time and is largely unsupervised. And then there's also an environmental problem to it, because NSAIDs, they can accumulate in the environment and pose a risk to aquatic systems. So, quick conclusion, we really need a solution that both addresses the environmental and the social concern. And this is where I start to talk about marine algae, because they are a source of bioactive compounds due to their ecological adaptations. And they offer a high potential for therapeutic development. So the University of Algarve set up a citizen science project called Algas na Praia, where they accumulated strandings of algae on the beaches of Algarve, and two algae here, Rugulopteryx okameri and Asparagopsis armata, were found to proliferate a lot in that region. So these algae were assessed in this thesis because of their highly available biomass in the region, because of their reported bioactivities in literature, and also additionally for Asparagopsis armata because it's included in the Algae for IBD project. So here I tested the gamidified extract. The main mission was to investigate bioactive properties of extracts from invasive macroalgae species to identify new drug candidates, both for prevention and management of CIDs and generally other human ailments. Practically, for okameri, the whole process started in the very beginning of biomass collection, processing and extraction. And for armata, I just tested the bioactivity itself, starting with cytotoxicity of extracts to the cells, and then also anti-inflammatory and antioxidant properties, as they are the two key factors in chronic inflammatory diseases. And these two bioactivities I assessed with in vitro cell line models. So let's talk about the methods then. A quick overview of the experimental design. As I said, for Rugulopteryx, we started very early in the process where biomass was collected, cleaned, dried, freeze-dried actually, and then also grinded into a fine powder, which was then assessed for the size of the distribution. And then the powder was extracted with EDGE, which is energy dispersive guided extraction, an automatic pressurized liquid extraction with 3 different solvents. After which the actual evaluation of bioactivities took place. For armata, as I said, it's a bit different. The extracts were received from Algae for IBD, the project, and here I'm just talking about 2 solvents, both from gamidified armata. So then the human cell-based assays, three main things, always starting with cell proliferation measurements. So this is the assessment of the toxicity of the extracts to the cells, measured with MTS colorimetric assay, and just measuring cell viability compared to a control without extract. Then for the antioxidant activity, I used CLPAA, which stands for cellular lipid peroxidation antioxidant assay. So it is a quantification of lipid peroxidation within cell membranes of cells, in this case, human keratinocyte cell line HACAT. And then the third part was anti-inflammatory activity. This is the biggest part of the thesis, and therefore two assays were used, a preventive one and a therapeutic one, and the preventive one is looking into the prevention of inflammation from occurring, and then the therapeutic one looks into the reversing or mitigation of already occurred inflammation. So more about anti-inflammatory then. I tested the assays on human THP1 macrophage cell line, and the assay was so that algae were exposed to human cells. Afterwards, the response of pro-inflammatory cytokines were measured. So this basically means the inhibition of cytokines, TNF-alpha and IL-1 beta, was quantified, which are anti-inflammatory markers after addition of inflammation. So it's very complex sentence, but you'll see in the graphs what that actually means. Then the second step is just the actual quantification with an ELISA in the conditioned medium from the cell assay itself. The assay timeline looks a little bit like this. We're talking about the preventive assay now, which starts with cell differentiation of THP1 into macrophage THP1 cells. Then the extracts are added to the cells, and then the inflammation is induced by adding LPS. And then after totally 48 hours of extract exposure to the cells, the condition, is this touch? No. Is quantified in the conditioned medium. The therapeutic, I continue, the therapeutic assay timeline looks exactly the same, with one difference, that the inflammation occurs before the addition of the extracts. So let's go finally to the results. I present to you first all the results of one species, Agmata, with a very simple graph showing the cell viability of THP1 cells. So we have here the cell viability and one control in blue, the MAC THP1 cells without the addition of any extracts. Then we have two solvents with different concentrations, and we can very clearly see that there's no difference in cell viability. These are results from two independent experiments with triplicates, and just to show you that we can go to the next step, which is then the anti-inflammatory activity, where we test the concentrations that were not toxic on the same cells. So this is a very complex graph, and let me take some time to explain how it works and then get into the data itself later. So we have TNF-alpha, which is indicator of inflammation. So a lot of TNF-alpha means a lot of inflammation. There are several controls. The first one being MAC THP1 cells without anything else, they're just cells. We have the same cells with the addition of LPS, which is stimulation of inflammation. So they're inflamed, you can see TNF-alpha rises to the sky. This is the positive inflammatory control, and we also have a positive anti-inflammatory control. This is dexamethasone. And you can see this is a very strong anti-inflammatory compound, drug actually, that in this case significantly reduces the TNF-alpha levels. Here the significance is indicated with stars, resulting from a two-way ANOVA with multiple comparisons, each time relative to the LPS, so the blue column. And also the A and B statistics are resulting from an unpaired t-test. So then, we have the result itself. At concentrations of 1 and 100 microgram per milliliter of the algae extracts, we see a downregulation of TNF-alpha levels which just means that there's less inflammation, so there's anti-inflammatory activity. We can even see at ethyl acetate extracts that at 100 microgram per milliliter, there's even a more significant decrease than the strong anti-inflammatory drug dexamethasone. So then, exactly the same graph with different cytokine I1 beta, just to show you a more dose-dependent results. Here we have the higher concentration of algae extract, the lower the inflammation to a not detected level. This is again the same graph, but we're talking about the therapeutic assay now, with in mind the more drug development. So we're talking about drug development or preventive, which is more like food additives, for instance. Here again, exactly the same results, a dose dependency, but also a higher significance, a lower TNF alpha levels compared to the highest concentrations of extracts compared to that of dexamethasone. And also for all concentrations, there's a significance here. Let me go to the antioxidant activity then. This is a completely different graph. We're showing inhibition of oxidation with a positive antioxidant control, quercetin. So it's a very strong antioxidant compound. And concentrations up to 100 microgram per milliliter show a not significant result compared to the quercetin here, which actually means that there's the same or comparative level of inhibition of oxidation. So conclusion here, aromata can potentially be antioxidants. So I finally come to Okamerai, which I again start with an cell viability assay, and there's a lot of columns here, but what is really important to look at is the concentration 5, because from here onwards we see a decrease in cell viability, which means this extract is toxic to THP1 cells. Considering these results, I also tested on different cells, Hakat cells, and here at a concentration of 5 microgram per milliliter, we don't see a decrease in cell viability, but an increase in proliferation actually. So this could potentially mean a selective toxicity. And here it is important to know that THP1 are actually cancer-like cells because they're isolated from leukemia patients. Then once more the inflammatory activity of Okamerai then, and here we can see at lower concentrations that were not toxic, a decrease in TNF alpha levels, so it's also anti-inflammatory. And for the antioxidant activity then, here's a completely different result because almost all of the values are negative, which means that they're actually inducing oxidation, except for all the lowest concentrations then. But this is not really a good value result, so here a very simple conclusion, Okamerai is probably not very antioxidant. Then I come to my conclusions, where I go back to the inflammatory part. So we saw 4 cell-based experiments of the extracts on LPS-stimulated MAC-THP1 cells. That for aromata, we saw a actually never shown anti-inflammatory potential, both in therapeutic and preventive context, that demonstrated dose dependency to reduce inflammation. And for Okamerai, actually the same thing, but it's just that we can only test lower concentrations, so this was really the main limitation. Nonetheless, we can see that it's compared results to the anti-inflammatory drug dexamethasone. So yeah, again, both algae show potential to be developed into both nutritional supplements, if you look at the therapeutic assay, but also can be developed into drugs if you look at the therapeutic assay. I think I said the same thing twice. Anyway, the selective toxicity then, as a second most important thing, we saw that Okamerai demonstrated selective toxicity inhibiting the growth of cancer-like THP1 cells, while also proliferating the ones of Hackett. So this is really interesting, can lead to some anti-cancerous properties. So this really has to be investigated further on different cell lines, for instance. For the antioxidant results, Okamerai did not demonstrate antioxidant activity at all, but aromata did. I have to add that these essays were more complex and there were more controls, so this really has to be on point to show highly reliable results. So my advice here is to just repeat and improve the essay itself. Then one more slide for the future perspectives. It will be very interesting to see the true potential of Okamarai, because now we have together in the extract toxic compounds, but we also have bioactive ones. So we could separate them with HPLC, fractionate, and then we can probably test higher concentrations of interesting compounds from the extract. And then also some context regarding the methods here. I tested using human cell lines. This includes some degree of complexity of the cells, but we're not talking about the whole organism. So in the future, it is important to test in vivo and also ultimately in clinical trials, which are a logical next step in the practical creation of any drugs or preventive agents. So this is where I conclude. And if you have any questions, please.
Thank you, Niels, and for almost respecting the time. Let's start with
Okay.
Alexander.
Thank you. Nice presentation, interesting results. Okay, I might have missed this, but what was the source of the seaweeds that you used for the extractions? Well, I assume the Okamura was something that you collected in the field.
Yes, exactly. So they were collected from an external corporation, an ex-student that is specialized in collecting fresh algae, but stranded on the beach. So they were collected in this case in Sagres in Portugal. For the Okamarai, of course.
Okay, for the Asparagopsis.
I don't know much background because the extracts came to the lab already made.
Okay.
So the origin is only disclosed to algae for IBD context.
Okay, well, I understand. No, I mean that's a quite important aspect.
It's a big difference between the two species. Yeah.
Biological activities of the macroalgal compounds depend quite a lot of the conditions in which the material
exactly,
is living.
I do want to say that they extract it in the same way. So it's also edge extraction under the same treatment.
Okay. No, I was wondering about this because in the case of the Rugolopteryx, if it was a material that was stranded, I mean the compounds and the activities that you recorded that might not reflect at least not fully the activities of the seaweeds when it is alive in its own environment, which okay, I wouldn't be able to tell if maybe it would be collecting the algae alive directly from the field
yeah, but they were
collected in this way would have more or less the activities of different types of the activities.
So for this batch of algae, they were collected freshly, frozen the day itself, and then they were freeze-dried. So they should be there should not be any degradation of compounds.
In the case of Asparagopsis, instead, at least do you know if this was something that was collected from the sea or they maybe obtained from cultures in the
I think it was collected in the field. I don't think it's cultivated.
No, because in the case of the Asparagopsis, there are several applied studies which have used actually the sporophyte, the Falkenbergia, so by cultivating it in the laboratory. Now, look at all this is very, very valuable. I see what is the point of the studies, trying to make a resource of something that, of course, in nature, and especially I know the story with the Rugolopteryx that now it is really a huge problem in that area. Look at do you think is there any chance that they will get to an exploitation
Well,
at the industrial level of the
of course,
species?
it's a long way, and it's definitely a challenge. But these results are quite interesting, and I think the most feasible thing to do is for now use them in food supplementation. But there's still a long way to go until creating something that actually can be used, I would say. But I definitely, I'm confident in the long term that these algae, especially Leucomyia, will serve for some purpose.
Look at another minor comment. Now, okay, in the title you mentioned invasive seaweeds. Now, in the case of Asparagopsis armata, I will no longer use the term invasive because, in fact, it's alien to Europe, but it has been around for more than a century. And other thing, I assume okay, for the purpose of the EOTs, say you didn't try to characterize the main compounds that are there.
No, so I used
just the test.
different solvents with the idea that some compounds might be more polar, and so you have an idea of what compounds you're actually extracting. But there's no significant difference between different solvents. And there was an idea to fractionize to actually isolate vitamin K because it's important in a lot of disease-related things. But for now, just the full extract was used. So,
Maybe coming back to you after. Yeah.
Thank you for the presentation. You for Asparagopsis, you have some very nice, interesting result on anti-inflammatory aspects, and
Yeah.
do you have explanation why it works?
why it works?
Yes. Okay, it works. Why you have positive result on cytokine, etc. But why?
Yeah. Well, therefore, I don't have any data to explain that, but therefore you have to look into literature that gives a whole list of compounds that are tested in biochemical assays that are anti-inflammatory. And it's really like, if you look at the review articles, it's a lot of potential reasons. But if you want a definite answer, I think you really have to just fractionate this algae and find out yourself. I don't know, because
I will do that.
it is written in literature. Well, yeah, I'm not gonna.
Do you with the literature and of what you do you formulate some hypotheses to explain that it's important to understand why it works, and because it's not like that, there is always a biological reason behind everything.
Yeah.
What is your point of view based on literature if you want?
It's a difficult question because I think about the pathways the compounds have, how they interact with these cells, and I don't know. I can't give you any answer about that. So I know I wrote in my thesis some how these algae work, how these compounds of the algae work. We know that they have those compounds, but I actually don't think I can specifically answer your question.
Okay, it's effectively not very easy to answer. What is your? It's quite fast and cheap to make in vitro assay. How confident you are when you, in your perspective, you open the door to say, okay, now we have to test in vivo level. What is how confident you are to have?
So
I say on Asparagopsis because you have very strong effects.
Yeah.
How do you see the next steps?
So to give you some context, the methods that I used, the setup of the anti-inflammatory activity is given by Algae4IBD. It's a project that scans a lot of algae, always in the same way to make sure they're very comparable to each other. So I didn't question these methods. And if you look at these results compared to other algae, they're very anti-inflammatory, especially in lower concentrations, which you never see, almost never see. I don't know all of them, of course, because I don't have that information. But I know from my supervisors, my colleagues, that it's a good result. compared to other algae.
How do you see the next steps to test? Sorry?
So, okay, that is, okay, it's quite efficient, but yes, effectively, are you very, what is your point of view to transfer in vitro assay to in vivo assay? It is always, you will observe exactly the same thing, so it will work, or do you have some doubt, or compare maybe with other things that was, because effectively for the in vitro assay it's very standardized, and after, how it will, what, yes.
I think I have an answer, I'm not sure. But I think it's a whole row of assessment. It starts with biochemical assessment of compounds, and then if you have an idea of what is interesting to test, you test in vitro than in vivo. And then the ones that are having a positive outcome of that assay go to the next step. So it's a screening because you can't test all algae on human cells. It just takes too much time. It's really expensive to do. And only the very few, very successful algae could then be used further in the whatever step comes next. I'm not sure if that is what you want to do.
Thank you.
Well, we have 4 minutes for questions.
Okay.
What about the dose that you used of algae? The dose, the concentration, the quantity of algae. Do you have any idea if it's something feasible for humans to ingest or otherwise put ourselves in contact with, so it could work?
Good question. But so the first step is the assessment of the cytotoxicity. So I can say that it's not toxic to these THP1 cells, and this is how far the conclusion can go. I can't extrapolate for.
No, I don't know if you have any of the quantity, like the biomass of algae it corresponds to.
The biomass of algae, yeah. So what I have is the extraction yield, which was very broadly around 10%. So if you have 1 gram of algae, you have 10 micrograms of.
Okay, so it's not that much algae I will have to ingest to get that concentration?
Well, I'm not sure if it works exactly like that, but
okay.
But generally speaking, yeah, that would be what it is.
Okay, can we go to the ethical aspects of working with human cells? Can you tell us a bit what you have to consider by using it?
Working with these cells is very general. There's a lot of people doing it, apparently. THP1 is the most standardized cell to use because they're very easily cultivated and very generally. So,
do you have specific requirements? Things you could not do with human cells that you could do with microalgae, for example?
No, I don't know, I'm not aware. The only thing I can say is that since these algae are invasive or not native, I should say now, that I had to be careful not to put them down the drain into the River-Mosa system and these kind of.
Not specifically with human cells. And just to make sure I understood it, several times you show that the dosage of the algae is not positively related to the effect on the inflammation.
Yeah, so for instance,
like
this graph,
if
right?
you increase the concentration of algae, I mean, it seems to be better. I don't know, what's this one?
Yeah, so this is how it looks most pretty, let's say. So we have a decrease with an increase of extract. Here we don't see that, but the standard deviation is quite high. So yeah, it's probably somewhere in the middle.
So
why?
You still say that there is a dose-dependent response. So it will mean with your results that we should aim more with 1 microgram than 10 microgram, for example.
Yeah, because what I actually want to emphasize here is that it actually doesn't even matter what the other concentrations are. What really matters is that at a low concentration, there's already a very significant downregulation.
Okay.
So what concentration you want to use, I don't know.
Big change, okay. From a low concentration.
But I think you should look more to the comparison to the LPS control, to the inflamed cells and the lower inflammation in the treatments. So.
Thank you for your short answers. It allows me to ask the last one.
Okay.
But you talked about citizen science?
Yeah.
So what's the need of citizen science in this process, more precisely?
The whole citizen science project was before my thesis, but I can tell you about it. So they set up this platform where people, beachgoers, tourists, can be lifeguards. All people can fill in a platform, send in pictures, and they use this data to map, if that's the correct word, the strandings of these algae. And then with the oceanographic conditions, they linked that to know where these algae thrives, proliferates, accumulates.
To get them? To collect them?
Not necessary to collect them.
Why not?
Well, yeah, why not? Because I think it's important to know that it's a very touristic region, so you don't want to have these huge piles of algae in front of your door. I guess that's a good reason to collect the
Next question. We have 30 seconds. So maybe food is already served, not available.
I was just wondering if for Ublopteryx at the moment there are any other possible usages that have been investigated or which you are aware of?
Oh. There's a lot of uses for algae, and it's a maze of applications for different algae species. And
thank you. That's all.
Sorry for the.
Okay, so see you for the next session at 2 p.m.